Okay. Uh, right. Let me just share my screen. So hello everybody. Like I just mentioned, we'll be beginning with the class. Uh, yeah. Was anybody trying to say anything? Okay, somebody's mic is turned on. Let me just check who. Uh, Deepika, I think you've just joined. Do you
Yeah,
want
yeah.
to? Yeah, yeah. Were you trying to say something? I
No,
couldn't hear
no.
that.
I'll just
Okay,
off my mic.
okay, sure. Okay. So in that case, I believe I'm still sharing the screen, but let me just share it once again, just so that we know that nothing has gone wrong over here. Yeah. Uh, okay. So hello guys, welcome. And this will be your final practice session for this particular course, that is Introduction to Professional Scientific Communication. Although sometimes we do have additional sessions just to for an overview of the entire practice session as well. Although we haven't scheduled one as of now, so if that will be getting scheduled in the coming week, NPTEL is going to inform that to you guys as well. For now, as per our knowledge, this is going to be our final practice session for this particular course. So let me introduce myself one last time for this course. I am Arveen Kaur. I am a PhD scholar at Department of Biological Sciences and Bioengineering at IIT Kanpur. And we are going to discuss in this particular session the previous year's assessment that was assigned for week 4 lectures, right? So, along with that, like I did in the previous meeting as well, I have added a few questions pertaining to what was discussed during this week's lectures, right? So, we had more number of lectures this week. I think there were 8, right? So pertaining to them, I have also prepared some additional questions for your practice. We are going to discuss them as well in this particular presentation only. Most of the materials that I have picked up for the same are from Professor Ganesh's NPTEL lectures, and wherever I have used another other sources, which I haven't much in this particular presentation. I have mentioned it. Also, uh, okay, so we will actually just begin with the lesson and then we can discuss during our questions, right? Okay, just a moment. Yeah. So our first question is, which of the following choices accurately describes the role of the journal editors who handle research papers submitted for review? So, is their role confidentiality or professionalism or data protection or all of these? I hope you remember who the editors of journals are. Whenever we submit a research paper, they are the first people on the behalf of the journal, or in that journal, they are one of the first people to actually see the manuscript that we have submitted, right? They are the ones who pass it on for review to other reviewers, to appropriate people from our field of research, right? So that they can review, they can give comments, and if any revisions are required at all, they can basically ask for those revisions, right? So the first people to look at our manuscripts are actually editors, and their responsibilities or uh, job requires all of these, right? So why do we say that confidentiality is required? Because whenever we are handing over our manuscript or for research paper, we have a few pages of information which withholds information as to what of whatever we have done for the past, say, uh, maybe 4 years, 5 years, maybe less, maybe even more, right? So whatever relevant data we had out of that hard work during those very, very, long years of your PhD, your postdoc, your dissertation, etc., that will be contained in those few papers, right? So the editor has to have that confidentiality as one of the qualities, or as one of the requirements, because they cannot pass it on to somebody who might use that data, right? Because our data is not published as yet, right? So if somebody else uses our data, they and this person might have better resources, or they might be a resourceful individual in general, so it could lead to undue maybe advantage, some kind of undue advantage, due to which our research could be taken over by some other group. So confidentiality is actually one big part, and a journal editor must have, some kind of ethics regarding this. The other is professionalism. Obviously, any person in power or at any job that you are doing, you need to be professional, right? So even if I'm teaching, up to some extent, there is has got to be some level of professionalism that comes, or that is reflected from the kind of presentation I am preparing, or the kind of presentation I am giving. So it should not be like I have to maybe record a 1 hour or 2 hour lecture, and I'm coming and sitting and just chatting with people. So that is just not professional, because that is not what we are required to do at our job. So there should be professionalism, not just in this case, in all jobs. Then there should be data protection. So whatever we discussed when we were discussing confidentiality, the same things apply over here as well. Right, because the data that we are handing over is a very, very sensitive piece of information, because it has not yet been published, and that is why it does not like it has not been credited to our knowledge, right? Or that we have done this work. We have not received the credit for it as yet publicly, right? So, to protect that information is also a part of the job of the editor, right? So, all of these over here was the correct option. Then comes question number two, wherein they have given an image. So they say the image given here represents an example of a figure prepared for a research paper, right? So one of the images A and the other is B, right? So one thing I would like to give a disclaimer of is that please don't go on the fact that B is a blurred image, right? That over here should not be a point of focus. We will look at what the point of focus should be, right? So here the two images A and B are connected with a dotted line, with dotted lines and a square in figure A, right? Which of the following options accurately describes the most likely explanation for such a representation? So the first option is the two images A and B are from two different sources, and the inset likely was drawn to highlight the similarity in the pattern of a granular structure. Okay. Option B is the inset identifies the actual size of the object when printed on the journal. Okay. Option C is the image on the right side is likely a higher magnification view of the inset shown on the left image. And D is all of these. So I will give you a moment to look at the image, to go through the options, and we are going to discuss the answer. So a hint for the same is, if you know, like if you have gone through research papers and if you have seen such kinds of images, I think it will be very easy for you to pick the correct option out of these. Also, even if you are familiar with what an inset is, or what this representation is, which is a very, very common way of representing images again in the papers, it will be very easy for you. But in case somebody is relatively new to research, has not read those many research papers, or your field, in your field, the research papers might not have those many, immunostaining or such representative images. In that case, we will tell you that the answer is the image on the right side, that is B, is likely a higher magnification view of the inset that is shown on the left image, right? So, why I said that please don't go on the fact that this one is blurry. Usually it is not, and that is not how it should be. But sometimes maybe in a image you are showing an entire organ, right? A zoomed out view of an entire organ. For example, a 4x magnification view or a 1x magnification view of the entire brain, wherein you can see that there are some spots and some staining that is visible, right? But you want to focus at some particular region in the brain where you want to show some key or characteristic kind of staining that is pertaining to that section, right? In that case, you will draw such a dotted structure or dotted square. Sometimes it is not dotted, sometimes it is just a square, and you will highlight it, right? You will like, it could be the same image, cropped version of the same image, or it could also be another image taken from the same section of the same area at a higher magnification, right? So either way, it can be represented in this way, and option C over here will be the correct answer for that. Now, question number 3 is, writing the discussion section is the most challenging part of a research paper. Some of the pointers that are often used to draft the discussion section are given below, not necessarily in the order of execution. Identify the option that correctly orders these steps. So the first is, define the significance of your discovery and explain its meaning. Then N is, contrast and compare the findings you obtained with those of other researchers that has been published. O is, explain the significance of work by discussing its applications, recommendations, and implications. And P is, describe the study's assumptions as well as its strengths and weaknesses. So which one of these do you think is the correct option, or is the correct order in which these steps should be performed? Okay, let us come to the answer, and then we can go through these statements in the correct order, right? So the answer is the first option, that is M, then N, then P, then O. So how are these steps? Let us just read them out in chronology. First, we should ideally define the significance of your study and explain its meaning, right? So once we are in the discussion section, first we have to tell the significance. Although in introduction you would have briefly hinted about it, but the most important significant thing that you would have done in your discussion is bring up a research question, right? Then you would have planned experiments, then objectives to fulfill your research question. And over here, you will just say what is the significance of that research question, right? After you discuss the significance of your discovery and also explain its meaning briefly, then you should contrast and compare the findings you obtained with those of other researchers that has the research that has been published, right? So over here, what does that mean? That, for example, I am working on, say, brain cancer, right? So, and I am working on some particular kind of maybe non-coding RNA that might have a role to play in brain cancer, right? It is a hypothetical situation. Now, once I am done with my set of experiments, I have some key findings. I will obviously report those, then I will discuss about them also. But uh, in my discussion, what should happen? Should I only discuss like that, okay, this is what I have done. These were the the experimental results that I got. They might mean this. And there are similar studies that have also reported such kinds of observations, right? That this group has already reported in other kinds of maybe brain tumors that this is what was happening in their case also. But that is not the only thing that should be done, right? Along with this, I should also say that if there is a group that had earlier worked on this, but they were not finding what I have found, I should also report their study that in their work, although they were not able to find this, but when I did this, I was able to find this, and this might be, and then I will give a hypothesis. If I don't have a reason or a rationale behind it, at least I can think about like how logically it could happen, and at least I can write that down, right? It is a hypothetical statement that I will be writing down in the discussion, right? So I will contrast and compare means uh, not just compare but also contrast. So maybe I am preparing some one kind of drug for a genetic disorder, right? Maybe some other group has some other kind of drug for that genetic disorder. So in my discussion, maybe I will include their finding also. That also in the existing research scenario, these are the other options that are available, and these are the groups that have worked on that. Along with that, this is one more candidate drug that I want to add to this list, right? So such things should always be taken care of. Then in chronology comes P. So after this, I will describe the study's assumptions as well as its strengths and weaknesses. So again, because you are contrasting and comparing, not just comparing, right? With similar studies, you are also comparing with competing studies, right? After that, you will also, like I said, there might be some things that might not match with some studies, but you will have to give hypothesis that this might happen, or your assumption, right? That we think that this might be the case, that is why we are getting these kinds of results. So those things you will have to obviously address after you have stated such things already, right? And you will also discuss both the strengths and the weaknesses of your study. So it will be very, very, arrogant of a researcher to think that, okay, this is the discovery I have gotten. It cannot be improved upon in any case, right? So there are always research gaps, right? That is how people are progressing that research by finding those research gaps. So some person may think in a particular direction, but maybe after reading some such report, somebody else finds some other direction, right? That is how research grows, and that is how a knowledge database also grows about a certain thing, right? So thus discussing weaknesses is also important, because that might also give maybe clue to somebody else that, okay, uh, we know that their study have addressed this points, but they were unable to verify or validate these things. So maybe we can look into that, and maybe we can design a study that looks into that, right? So that is also how research will progress. Then last comes explaining the significance of your work by discussing its application, recommendation, and implication. So this is what you will do in the end, right? So having said that, we'll move to question number 4, which says science journals frequently require authors to certify that the submitted manuscript is not part of a simultaneous submission. Here, simultaneous submission refers to which one of these? The same set of authors simultaneously submitting two or more papers, each describing different work, or the same set of authors simultaneously submitting the same work or results to two or more journals, or simultaneous submission of almost identical findings to the same journal by two independent groups, or any of these. So this is something that Sir did explain briefly while he was talking about some ethics that we have to take care of while we are submitting papers, right? So let us look at what simultaneous submission refers to, right? So it basically means that the same set of authors should not simultaneously submit the same work or results to two or more journals, right? So you have to be exclusive, right? Although it is uncertain whether a journal will accept your work or reject it or send it for revision, at one time you should target only one journal, right? Because it might be the case that both journals accept your work and now both of them can't publish it, right? And once you are actually submitting your manuscript, you are agreeing that okay, this same manuscript or this same work I am not giving to any other journal, right? Only then will the reviewers of one particular journal look at it, because reviewers are also scientists, right? So they, and it is not their only job to scientists have to do science, they have to do experiments, or they have to look after their students or their lab members who are doing experiments, right? They have to guide them. It is not their sole job on earth to just read papers and to review papers, right? So if somebody is doing that, it is ethically wrong that you are making two sets of people do the same thing for the same paper, and they might have very similar concerns also regarding the paper on which you will work, maybe. And there are a lot of people whose time or and energy and you know a very valuable time you will be wasting by doing this thing, and it is like ethically not correct, right? Even if you are maybe a backend coder for one of the websites, right? For example, for a medicine delivery website, if you are making a user interface, maybe not backend, maybe you are making a user interface or designing or coding a user interface for a medicine delivery website, right? And you go and the same code you also sell to another medicine delivery website. So that is not ethically right, right? They might do the same kind of function, but you cannot just copy the work you have done over here and go and paste it to the other company, right? Because this first company invested money in you so that you will design something for them, and you can not just say that I was the one who designed it. Now I can go and I can sell it to somebody else also. Right. So for that one job you did, you cannot expect two people to pay you. Right. So ethics are kind of like that. Okay. So question number 5 is, science journals often obtain publishing rights from authors before publishing the work of the authors in the journal. Which one of the following options is not true with regard to publishing rights transferred to the journal? Okay, so preventing the authors from republishing data and figures on commercial websites or in books, preventing the authors from republishing their data and figures in thesis and lab reports, preventing the authors from publicly sharing the published articles on their website and another similar public forum, preventing authors from claiming copyright for the text and images after article has been published in journal. So please go through these options. Okay, I believe I have marked the option for this incorrectly. Let us see what I, yeah. So the question says which one of the following options is not true with regard to the publishing rights transferred to the journal, right? So when somebody is asking, the science journals are asking for publishing rights from authors, they do require that authors don't republish the data and figures on commercial websites or in books, right? So this is the incorrect option that I have marked. The correct option will be preventing the authors from republishing their data and figures in thesis and lab reports. So thesis is something that you are going to submit to your institute. It will contain all the data and all the figures that you have compiled in your tenure as a master student, as a PhD student, etc. Right? So this is one place wherein I the journal will not have these publishing rights, right? Because you will, for just for a thesis, you will not perform a separate set of experiments, right? Whatever set of experiments you have performed will go in your thesis also, and that a part of your thesis can be published in a paper, right? So this is actually the correct option. All others, that is preventing authors from republishing data and figures on commercial websites or in books, preventing authors from publicly sharing published articles on their website and another similar public forum, uh, is also not true actually. Yeah, we'll just revisit this question, okay? Because this was the data that this was the question that was this was the option that was marked as the correct option in the previous year's assignment. I have marked it the same. I am having some second thoughts on it, so we will revisit this after a while. We'll just get back to it. For now, let us go through question number 6, which says a group of students created a slide presentation to explain gluten, a protein naturally found in some grains, right? Examine the slide and select the appropriate critic from the options that are provided below. So this is the slide, right? The one I am going about my pointer with. This is a slide which says what is gluten. Then there is point number 1, 2, 3, and 4 that are given, right? About gluten. So these are the critics that are provided. Which one of these is the appropriate critic for this particular slide, right? So critic number 1 is too dark background image, which does not add much information to the content, right? B says too many colors for the text material do not add any value. C is color contrast of the fonts with the background are hard to read, right? And D is all of these. So I think this one was a simple one because it is a very, very bold background over here. And it is very, even if you try to read the points, you will understand that it is really hard to read those points. Also, the colors that are provided, as you might remember from sir's, this lectures, that usually when you are using different colors, they mean something, right? So maybe you are trying to segregate some discussion. You are using the same slide for two kinds of discussion simultaneously. In that, if you are using different colors for these two sides, it makes sense, right? You are trying to look at them differently. But if unnecessarily you are just bringing in color and that doesn't mean anything, it might confuse the audience a little, right? Also, you might remember from his lectures about the color contrast, right? For example, on a white background, if we write with a dark color, this black, blue, red, all these bright colors, it will be visible. But if I choose to write on this very light background with a, light pink color, a yellow color, uh, it will not very well be visible, right? Or maybe on a black maybe background, if I'm writing with a gray color, it will not be very well visible, right? So you have to look at the color contrast of whatever you are writing as opposed to the background that you are using. Okay. Then we come to question number 7, which says the majority of the papers published in scientific journals contain a section titled Acknowledgements, in which the authors recognize the contributions of the individuals or organizations. Which of the following characteristics should not ideally be included in the acknowledgement section? So it is a repeated question from week 3 assessment also, but we will go through it. The sponsor who funded the research project, an expert who read through the manuscript and provided critical input on the content, a colleague who shared some research reagents for the study being written, or a friend of an author who helped him financially during his elementary school days. Right? So, which one of these should be the correct option? Since we have already discussed this in the last session, we will again highlight that this is the correct option. And we have at length discussed in the previous video also why exactly this should ideally not be included in the acknowledgements. This time I have also brought in a screenshot from Springer Nature's, kind of guide as to how a paper should be written. So in Springer, in the acknowledgement section, what they have suggested is thank all of the people who helped with the research but did not qualify for authorship, right? Those who provided intellectual assistance, technical help, or special equipment or materials, funding body or for research grants if applicable, right? So over here, yeah, Vikas, you can ask.
Ma'am, please go ahead with the discussion of this question. Actually, I have something to discuss in previous question, question number 6. Once you complete this one, can we go back to question
Yeah,
number
yeah, yeah,
6?
sure, sure. Okay. So actually that was it for this particular question, that over here, why are we saying that ideally this D option should not be included in the acknowledgements is because what the journals themselves suggest is that we will include those people in acknowledgements who are directly involved with the research, right? Some area pertaining to the research, whether it is funding the research or providing reagents or providing, some motivation pertaining to research, some technical help, etc. So that is who we should ideally acknowledge in our paper. Okay. With that said, we can actually go to this. Yes. Yeah,
Ma'am,
Vikas.
here point B and C options seems to be very much satisfying, but option A, what it tells is that too dark background image, which does not add much information to the content. Here, when it is referring to the
Mhm.
background image, the background image is not at all too dark. We can see obviously it is not dark image. So I am not convinced with why this A should also be included in the option.
It is relatively actually a dark image because if you have gone to these options wherein you pick an image and you put it as a background, there is an option of transparency in that, right? First of all, it is not, suggested. You can keep an image as your background, but a busy image as this one could maybe be your background in your, uh, end slide, the thanks slide, or in your first slide where you are introducing the topic. Firstly, whatever slides have content that is written on them should not have a very, very, busy background in any case. Like for example, if you are you aware with the concept of watermarks?
Yes, yes, madam.
So just imagine that if this screen that you are seeing, with the one that I am sharing with you, right? If this screen was filled with watermarks, right? To an extent, like a watermark maybe going through the middle or on one side, etc., is not much of will not make much of a difference. You will still be able to read the content, but if there was a continuous watermark that would be spanning the entire presentation, right? Maybe something saying Arveen Kaur, NPTEL, then today's date and the course name, and that on loop repeated, right? that being present in the background, will you be able to read what this question is or what these options are very nicely? So I expect not actually, right? So that is why the background should generally be neutral, right? Even if there is some such background being added, I have added such backgrounds, but then I don't add it on the report section where I am including results or where I am writing something, right? I add them in the title slide, wherein there is just one or two lines that have to be said, and the background can give much more information. That okay, for example, I am talking about lichens in this particular presentation, so I have kept a very good and very visually stimulating image of a lichen in the title slide, right, which says lichens and whatever the subtitle is and my name maybe, right? uh, but once I come on to what are lichens, like how are they, what are the constituents of lichens, right? How does fungus contribute to the existence of lichens? How does algae contribute to its existence? Over there, it will not make sense, right? Because it will be hard for the person who I'm presenting it to read it in the first case. For example, let me just exit this for 1 second and maybe I don't know where the watermarks are. Anyway, we will just see. Mm, Let us try to find such image. Yeah, I was talking about some such image, right? Oh, I've opened Quora with no this thing. Oh, okay, sorry for the interruption, guys. I am just trying to show something. You see, when your background is very busy, it will be very hard for you or tough for you to actually see what is written also. Right. So just because of that reason, we are considering option A over here as well. Right? That the background indeed is too dark and it is not adding much information, because what is gluten that we are understanding from what is written, right? Just by seeing that, by seeing the image, what can I make out? That okay, if I'm aware of it, I can see that there is something like wheat or barley that is lying in the background, right? I don't know what gluten has to do with it, but maybe it has something to do with these particular things. But if I read the text, what will I understand? That gluten is what is given in the text? Gluten is a naturally occurring protein in wheat, rye, barley, and triticale. So, okay, this I could somehow understand from this image that it has something to do with these, pulses or wheat or whatever is given over here. But I could certainly not figure out that it is a protein. Right. Then the gluten protein is formed by combining two molecules, glutenin and gliadin, that when mixed with water create a structural network, right? This is also something that that image could not tell me. Then the other two as well, right? uh, Oats one is particularly very difficult to read. So, because the text is also very light of a very different color. Anyway, that is why we have actually included this as one of the options, wherein the background is not really, adding any relevant information over here. And any slide with content in it, like written content in it, should ideally not have a busy background. That is the only point. Okay. Okay. So I think we can go on to question number 8 then. Let me just move my screen. Yeah. So question number 8 states, following is a statement with two sections that are denoted by blue and red color fonts, right? Examine the statement and select the option that is true with regard to the guidelines followed in a research paper or project, right? Let us read these statements. A research publication frequently adheres to a standard format for citations, and such formats ensure that ethical guidelines are adhered to correctly. Okay. So our options are both the segments or both these statements in blue and red are correct. Both the segments are incorrect. Segment shown in blue is correct and the segment shown in red is incorrect. Or segment shown in blue is incorrect and segment shown in blue is correct. In red it should be. In the second statement will be segment shown in blue is incorrect and segment shown in red is correct. Right? So which one of these do you think is the correct option? I have just zoomed out of it. I realized that a while later. Yes, Vikas. Okay.
Yeah, it should be option C, yes.
Yeah. Okay. I have just made that correction so that I don't hand out the, this typo thing to you guys when I'm uploading the material. Okay. Yeah, so indeed, segment blue, whatever it is saying is correct. But does what segment red is saying make any sense? So what segment blue is saying is research publication frequently adheres to a standard format for citations, right? These formats could differ from one particular publication to another publication, right? Some of them want something in maybe APA format, some other want in some other kind of format, right? So these are just formatting or structuring, right? For example, I want it in this font. I want the author's name to come first, or I want the title to come first, or I want the journal to come first, right? While you are citing. So there are different kinds of formats that are there, and it varies from publication to publication. So the blue segment is absolutely correct. But the red segment says that such formats ensure ethical guidelines are adhered to correctly. No, so that is incorrect because just the formatting cannot make a person adhere to ethical guidelines, right? What is making them adhere to ethical guidelines? Citing it in the first place, right? Just the formatting of that citing will not help in, adhering to any kind of ethical guidelines. That whether this title will come first, whether this author's name will come first, that will happen once you are actually citing a paper, right? So this is incorrect. So with that, we'll move on to question number 9. Question number 9 says, which of the following is not an effective method for displaying the overall trend of a large data set? Right? So the options are bar diagrams, pie charts, tables, or line diagrams. So think about it. What if you have a large data set? Yeah, Vikas.
It must be tables.
Yeah.
Other options satisfies the requirement of large data set representation.
Yes. That in fact is correct. Because when we talk about components such as line diagrams, bar diagrams, and even pie charts, there is a very they're very flexible in the terms that one point could be at maybe 0 or in tens, right? Something 20. The other point could be as high up as maybe 20,000, right? So, or 2 lakh, right? So that much flexibility is there when you are depicting data via bar diagrams, pie charts, line diagrams, right? But when you are depicting something via a table, right, you only have so many columns. So you cannot just, if you have a very large data set, it will not be feasible for you to just keep making those entries in columns, right? If that would have been the ideal case, we would just have, drawn these tables, and Excel would never have developed to, do like whatever entries we make in an Excel sheet, we would just have submitted those, right? The Excel would never have been modified to be able to make diagrams or pie charts, etc., so that whatever these long data that we are entering can just come as one particular image, right? So, tables in fact is the correct option over here. Which brings us to the final question for the previous year's assignment. We still have more questions to go, but in the previous year's assignment, this is the final question for the segment, and it is: a group of students created a novel strain of an infectious fungus whose spores caused skin irritation when they came into contact with human skin. Students wish to determine if the newly developed virus was infectious. Based on your knowledge of research ethics, choose the committee that needs not to be approached for permission to conduct the proposed research. A is Institutional Ethics Committee for research involving human subjects. B is Institutional Animal Ethics Committee. C is Institutional Biosafety Committee. And D is none of these. Yes, Vikas.
So it must be option A
Okay.
because it is
But
involving humans experiment here.
the question says needs not to be approached for permission. Would you like to change your answer?
Yeah, then it must be none of these because the committee which should approve this one is A. Other committees have nothing to do with this, specifically when the human subjects are involved. It should be none of these then.
So over here, what we have chosen is Institutional Animal Ethics Committee. I will tell you why. Because over here, the students that are hypothetically working on some kind of fungus which is directly coming in contact with or causing some kind of infection to humans, right? And they are proposing a research to study more about that infection on humans only. So that is why the first thing, that is the ethics committee for research involving human subjects, has to be approached, and their clearance is very very important because humans are involved, and this is a committee that is dealing with the uh, approving this involvement. So this committee definitely has to be involved. Then institutional biosafety committee does what? So for example, there are different for materials that we use. There are a lot of chemicals that we used, and a lot of them could be hazardous, right? Hazardous to us if we come in contact with them, right? Without having used gloves or without any protective, this external uh, external protective gear. If we come in contact with them, they could be hazardous to us, to the animals around us, to the plants around us, etc., right? So if somebody is working with an infectious virus, that obviously could be hazardous to humans and to other, maybe it might be hazardous to some other, animals or some other people that are coming in contact with these particular group of researchers, etc. So institutional biosafety committee does have to be approached. What this committee will do is they will ask you that if this is an infectious virus, ideally what is the kind of biosafety level that you should have in your particular department or in your lab, right? So the researchers might say that we need BSL 3 or BSL 4, right? So there are biosafety levels, right? If you are working with something that is not at all infectious, you will work with biosafety level 1, which might have been infectious, but now it is not, right? It has been uh, kind of the infectious property has been removed. You might work with the biosafety level 2, right? So for this particular case, biosafety level 3 or 4, which 4 is the highest, might be required, right? So what this biosafety committee will ask is, does your department or does your lab have the provision for a biosafety level 3 or have a provision for biosafety level 4? In that, I will respond, if I don't have that provision, I will say that no, I do not have that as of now. So this committee will say that, okay, in that case, your proposal is rejected and you cannot work on this, right? Because it is not safe for you and for the people around you that you work on an infectious virus without having that kind of safety level incubators and safety level doors and infrastructure in your department, right? Because then everybody in the department might be at risk. So they will just reject my proposal. On the contrary, what they can say is that, okay, we will consider approving your proposal if you go and you get this biosafety level 3 established in your department, right? So after that, I will not work on this project. First, I will work on having that established in my department, right? I will approach the funding agencies. I will write grants so that I can get money for establishing that particular, biosafety level infrastructure in my department. After that, I will revisit the project. I will again submit another proposal, and only if every such thing is dealt with properly, this institutional biosafety committee will go ahead and they will say, okay. So if the ethics committee for research involving human subjects has no issues with you working on humans, since all your biosafety things have been taken care of, we also don't have any problem. You can go ahead with your proposal or your project, right? So these are the two committees that definitely have to be involved in this. The one that does not need to be, approached for permission over here for this case is the Institutional Animal Ethics Committee. When does this particular committee need to be approached? When you are working with model organisms, when you are working with some kind of animal, right? For example, I'm working with mice. So our institute has an animal ethics committee, and there is a animal ethics committee at a nodal level, at a national level also. And since I'm working with mice, I report back to them on a 6 month basis, on a yearly basis, that what is the status of the mice? Are they well? Is their condition okay, or are they getting some kind of a disease, right? Or are they dying, or are they being maintained at a good, in a good environment or not? Or they can come and they can check, right, for that approval, after the approval has been given, whether I am maintaining my mice properly, whether I am ethical, that not 20 mice should not be in the same cage, right? It will be very, very suffocating for them also. So if for example the suggested number is 8 or 9, so not more than 8, not more than 9 mice should be there in one particular cage. So all these things they can come and they can check, right? If that would have been the case, or if I would have to breed more animals to do my research on them, in those cases, how many animals do I want for my research? How many matings I will have to set for those many animals, right? How many animals will I actually sacrifice? When and for what experiment I will use their harvested organs, or whatever other things that I'm planning. For all those things, animal ethics committee needs to be approached. But over here, we are directly proposing to work with humans and to observe humans, and whether they are getting this infection or not. This committee can be bypassed and we don't really need their approval, right? So that is why the correct option for question number 10 was B. That is, institutional animal ethics committee can actually be skipped in this case. Okay, that brings us to question number 11, which says, which of the following should not be given in a result section? Okay, so inference of the results, observations, discussion about the observations, implications of the data, or statement about what has been found, right? So which one of these should be included in a result section, and more importantly, which one should not be included in a in a result section? So go through the options, whether it is A, B, and C, or C, D, and E, or B, D, and E, or A, C, and D. So go through these options and we will discuss. Yes, Vikas, you can say.
I think it should be option D because observations, that is option B, and the statement about what has been found should be there. So what should not be there is the remaining things, that is A, C, and D. That is what my understanding says.
Yeah. So you are actually right in that. Uh, and you very correctly pointed it out that observations, right? Whatever experiment you have done, you will obviously give that experiment, whatever observation you have gotten from that experiment. Visually, also, you will give those visual data also, and you will write it down also that, okay, this was the experiment performed. In my normal wild type mouse, I found this. In my knockout mouse or in my transgenic mouse, I found this, right? But I will not interpret these observations at that time, right? I will not say that I think that this might be due to this particular thing, right? That I will do in my discussion section, right? So I will not discuss my observation. I will not infer the results, right? That I think that because we have received this result, so this might mean that the mouse might have some kind of cardiac issue, right? This all these things I will do in my discussion section. And implications of the data, that since we have obtained this as the results in what in our experiments, this will mean that again it will imply something. It will mean that the tumor in the mouse was much severe when this particular drug was given, but the relapse was much less when this particular drug was given, right? So I will not do that over here. That will be again be saved for the discussion section. Over here I will just make the observations of the results that I have of the experiments that I have performed, and I will make a statement about what has been found. I will say that in when the mouse was treated, when the same strain mice were treated with this particular drug, there was more relapse. When they were treated with this particular drug, the relapse reduced, right? That is what I know. I am seeing it from maybe a PET scan, or maybe from some other kind of data that I have. I am just giving you that data and I am reading out what that, what I am seeing. I am not saying that this will mean that this drug should maybe it might suppress this particular pathway, and that is why there is more survival that is happening. Yeah, Deepika, you raise your hand.
I was saying that inference of results will then come in the discussion section,
Yeah, it will come in the discussion section.
and statement about what has been found will be result, and
Yeah,
implications of data also will be in discussion section only.
yes, it will be in the discussion section. That is when you will say that with all this data that I have gotten, uh, it will mean that my hypothesis that I initially said was true, it was untrue, or this part of my hypothesis was true, but we will have to modify, the treatment strategies further, this, that, something like that, you will say, right? So implications about data will also come in the discussion section, yes. Because in results, how are you writing the result section? You will obviously divide it into subheadings, right? One will be that maybe if you follow a flow of a paper, that is maybe looking for whether the drugs that are already existing in the market are working or not. Okay, this is the objective of that paper. So in your first experiment, what you will do is you will maybe induce and it is a cancer drug. Okay, so in your first set of experiment you will induce cancer in your model organisms, right? In that the result that you will write, or the image that you will show, is that okay, cancer is there in all these mice, right? And then I am beginning with my research. In the second one, you will give maybe 0.5 molar concentration of each of these drugs that you have shortlisted to work on, right? Then you will write that after I have given this is the observation that I saw, that okay, there was shrinkage and tumor in this one and this one, but all these others did not show any much of a difference, right? After 4 hours of treatment, maybe. After that, you will maybe treat it for, say, 3 months. After 3 months, you say that although initially these drugs work better, but now I can see that these other drugs in the long run are working better than these first two that started working, early on. Right. After that, you might, you know, take that mouse after it has been treated with everything. Now you say that you will check whether it has cancer or not, and say that none of these mice now have cancer, right? After that, you will validate this. Okay, so we see that after treating these mice for 10 months with all these drugs, like different drugs, different mice, we see that none of these mice now have cancer, right? After that, you will let these mice grow, right? Induce cancer in them. Another set of mice, you will induce cancer in them. You will treat them for 10 months. Cancer will disappear probably. And after that, you will still not sacrifice these mice. After that, you will check or study these mice after 2 years. So that was in 10 months you had studied. After 2 years, you see you do a PET scan for the marker of the particular cancer that you are studying, and you see that, okay, in the mice that were treated with this A, C, and D kind of drug, the we see that the cancer is relapsing, but this B and E kind of drug, that has worked well because there's no relapse, right? Then you will write this. But in all this, uh, you have not inferred, anything out of your result. You have written that, okay, in this experiment, this is my result, and you will make a statement about it that when this experiment was performed, this was found. When this experiment was performed, this was found, right? Your observation and your statement about what has been observed, right? But all together, are you inferencing it together in the result section? No, because in my final one, for example, in PET scan, I will say that I see in that particular one, what I will write is that I see that in A, C, and D, there is cancer that is present. Cancer markers are showing up, but in B and E1, the cancer marker is not showing up, right? So that is my result for this one. After that, in the discussion, I will write that earlier these drugs, although these drugs show a better effect early on, but with the later with the chronic treatment with these drugs, it was seen that these other drugs were better for treatment, right? Out of these other drugs also, there were these two drugs which did not show any relapse of cancer even after 1 year of treatment, while these other drugs showed some kind of relapse. So all this you will arrange and you will tell it like a story is when you are discussing, right? You will not do it in your result section.
Thanks, ma'am.
Okay. So question number 12. Which of the following is an incorrect statement? Okay. So subheadings should be used to separate the results of different experiments, or results should be presented in the order in which the experiments were performed, or data should not be duplicated or repeated in text, tables, and figures, and results of statistical analysis should be included in text. So which one of these do you think is the incorrect statement?
Would it be B, ma'am?
Yeah. So it is in fact B. Because all these other statements that I read out are all correct, right? That it is in fact the case that subheadings are used for different, results in for different experiments. then it is the case that data should not be duplicated. Like, why will you write the same thing in a table and the same thing again in text, right? Or you should not use multiple illustrations for the same exact data in one paper, right? Then results of statistical analysis should be included in text, because whatever, for example, you have drawn a bar graph for a very large data set, right? But you need to state in your result section that in that bar graph, what is it that you are seeing, right? Because sometimes it might be possible that the thing that you are trying to show using that bar graph is not clear to somebody who is actually seeing that graph. But maybe once they read what you have written about it, they will be able to understand, oh, this is what he was trying to plot, or this is what she was trying to, show as a trend, right? But results should not be presented in order in which the experiments were performed. They should instead be presented in the most logical order, right? Uh, it might be possible that I start my project with some other thing in mind, right? But as I go through my project, I have some findings due to which I perform some additional experiments. Now, these additional experiments that were performed much later in my, tenure as a PhD student might make more sense when I present them first in my paper, right? That okay, I saw that maybe I'm working on mice right now, but sometime in the future, I will, stumble upon something and I will think that, okay, maybe I can work on this neural cell line to look at this, right? Maybe when I'm writing a paper, it will make more sense that first I say that in the cell lines, I performed these experiments. Based on that, this was the question. After that, I performed this experiment in the mice. Based on that, this was the question that was there. For that, I performed this set of experiment. So it might not be presented in the order that it was performed, but in the order that it is most easy to be understood by whoever is reading that paper, right? Because all in all, I have done that work, right? Whichever form I am able to present it, so that you are convinced as a reader that okay, what I am saying is actually true, right? I have the data for it, right? The data can be jumbled and it will still be the same data, but will I be able to get it across the same? That is a question, right? Okay. So, question number 13 is results section should include which one of these? A short paragraph of conclusion which talks about key findings, a table showing summary of all statistical analysis performed, inference of data and its possible implications for future research, or none of these. This is a tricky one because of the discussion we've had earlier. That is my only hint for this one.
Could it be none of these?
Which is why I said it is a tricky one. Okay. So it should ideally include a short conclusion which lists the key finding, right? Why? Because over here, although we are not inferring the data, but we have multiple subheadings, right? So that the experiments don't entirely seem detached at the end of it, and you might be publishing in a very data intensive kind of journal, right? From all these experiments, the one that is supporting your hypothesis the most, maybe you should get a chance to highlight that experiment, right? Maybe it has come in the middle of your this order of presentation, or maybe it has come at the end, or maybe it was the first experiment that you talked about. You should just write it down that okay, because of this particular I like, it will vary. It is very contextual of how you will write it down, but you will basically make a note of it, right? That because we saw that uh, So, for example, in the example that I was giving earlier, that cancer drug example, right? The short note for my key finding could be it is the last experiment that I performed, right? So, what will be my short conclusion paragraph? That uh, from this results, in the results, we observed that B and E could be the best candidate drugs for this particular kind of cancer. Right now, we have not explained uh, like why it is so, or a better statement for the result section could be that in this particular kind of cancer, drug B and drug E showed the best results when we worked with mice, because uh, mice that were given this particular drug did not show a relapse of cancer. Right. This could be a concluding statement for my result section. In this, I have not discussed anything that why is it so, right? I have not unnecessarily made inferences from my data, but I have just given that, okay, this is the key finding that I find from this experiment, right? For example, if you are doing a neuroscience kind of paper, right? And you are working on neurodegeneration. In that, you have established or characterized one particular transgenic model of mouse, right? So you have done n number of experiments on that mouse. On molecular level, also you've done experiments. You've done behavioral experiment with the transgenic mice and wild type mice. But in your key findings, what you will say that it is seen that uh, since the transgenic mice could not perform these behavioral tests well, it is possible that they do show memory deficits, which is also supported by this molecular experiment that was performed. Okay, that is all. Now, why this particular thing happened, we will discuss in the discussion section. That is why over here, this is actually the correct option. When we come to the recap for the previous year, this SIRS lectures, I have to cover that also in today's class for this week, right? Week 4, when we do that recap, you remember the statement. It is something I have picked directly from SIRS lectures. Okay. So question number 14 is, which of these statements is true with regard to non-textual element in your research paper, right? Each non-textual element must have a caption consisting of a number and a title. The number of figures, tables, graphs, etc. in the text should be should explicitly reference the item. If a non-textual element is adapted from some existing work, the said work should be cited. Or all of these. Okay. So the answer for this one is actually all of these. Uh, so the statements that I read out were all true with regard to non-textual elements. Okay. When we are talking about these elements, obviously they should have a number and a title. For example, if I'm talking about a table, so it should be table 1, table 2, table 3, right? If I'm talking about a figure, then figure 1, 2, 3, or figure A, B, C, whatever, right? Then the number of these figures, tables, graphs, etc. in the text should be explicitly reference item. Why at all are we numbering it? Because, for example, I'm writing something, I cannot always, paste what I am writing about directly in front of that, right? That will depend on the formatting of the paper, which I am not doing, right? That is being done by the journal only. So maybe I'm talking about something that the journal has, printed 2 pages ahead of when I am talking about it, or 2 pages before when I am talking about it. So when I'm talking about this, that okay, in this particular graph, in figure 6A, yeah, so we know that we have to go and look at figure 6, right? Maybe adjacent to when I am where it is being written, there is figure 7. So I don't have to go and look at figure 7. Figure 7A may also have a graph. But the graph that is being talked about in that text is the graph that is present in figure 6, subsection A, right? So that is why it should definitely explicitly refer the item. And if not, if you are adapting some of the work from, for example, if you are making a schematic, right? You might have made that schematic, but a similar schematic might have existed somewhere and you might have copied it and maybe made some tweaks, right? In that case, it should ethically be done that wherever you have adapted that from should be cited. Or maybe you have opened some textbook of cell biology and you are working on a paper with maybe centrosomes, right? You have picked up a cell biology textbook, or you are working with a cell division kind of paper, right? You have picked up Watson or you have picked up Lotus and you have opened that cell division cycle that is present in this, and you have kind of made it on your own or on your screen, but you have exactly looked at that and made one, right? So this means you have adapted it from that book, right? So you should always give reference to that particular book. Okay, question number 15. Why are figures and tables used in a research paper, right? Is it to enhance the color of contrast of the paper? Or the research papers without display elements are not accepted in reputed journals? Or they are the quickest way to communicate large amounts of complex information that would be complicated to explain in text? Or all of these?
C, ma'am.
Yeah. So C is the correct option. We are not adding, these figures and tables to beautify the paper, or our paper might just have a hypothesis that we are talking about. In that case, if the journal accepts papers that talk about hypothesis, they are going to accept it, even if we are not putting any, figures or tables, right? So that is also not the case, B. Right. So C is actually the correct answer, which is that when we talk about these display elements, they are sometimes the easiest way to communicate large amounts of complex information. For example, if you are proposing some pathway, some signaling pathway, right? You will write an entire paragraph that I think that this particular ligand will come and bind to this particular membrane receptor. After that, this particular membrane receptor will go and dimerize with the its counterpart. And after they have dimerized, then the phosphorylation of this particular thing will happen. After that, this signaling cascade. It is a very, very long statement to make, right? You can just make one particular schematic and it will show everything, right? So sometimes that is why you are adding figures and tables into your research paper, right? Question number 16 is, which one of these are a part of ethics in image processing for publications, right? The figures must maintain their original proportions. The specific features within an image may be enhanced, obscured, moved, removed, or introduced. The lanes of gels must not be spliced together. One particular part of image should not be at discussing, focused at while discussing the image.
B.
B is actually not the correct option. The correct option is that for image processing, there are particular ethics, one of which is that figures must maintain their original proportions. If you remember Sir's lecture in which he showed that mouse, if you are like literally increasing the size of the image or vertically increasing the size of the image, the kind of image that you are seeing for that mouse is changing. Right now, it will become very elongated or it will become very broad, oval kind of thing. Right? That is not what you want. When you are processing an image, you want that the figures should maintain their original proportions. Right? Along with that, it is said that you should not, obscure or enhance or move or remove or introduce a specific feature within an image. Why do we say that? If you are doing it to an entire image, it is okay, right? You can enhance an entire image, right? You can change the contrast of an entire image so that you can see the color that you wanted properly. You can decrease the noise of the image, etc. Right? But to a specific feature, you cannot remove some portion of the image, or you cannot introduce maybe in the image dots at some places were not present, and now you have included dots which look like the kind of data that is already present in that image. So you have tampered with that image there. So that is something you should not do, right? Then C option was lanes of gels must not be spliced together. So ideally they should not be, but it is not a rule. So the rule actually is that if you are actually putting these lanes together, you should write it that okay, I ran a gel, it ran diagonally. I don't know for whatever reason it ran diagonally. That is why these lanes were not matching the first three lanes. So I am put them together. I have spliced them, or I have put them together, with Photoshop or with some software. But you are writing it down, right? Or you are including some a line over there, which means that okay, these first three are coming from one part of the gel and these last three are coming from another part of the gel that has been uh, manipulated and moved a little forward, because it might happen sometimes that the gel, it doesn't run in a straight line, right? So for whatever reason, then one particular part of the image should not be focused at while discussing. Now, this is not true, right? We were talking about insets earlier in this discussion, right? In that case, you cannot tamper with one particular part of an image, but you can focus at that part and you can discuss about it, right? So that is also not true. So the only true or correct statement about image ethics in image processing was the first one, that is figures must maintain their original proportions, right? Coming to question number 17, which says which of these points should be kept in mind while citing papers? A is we should cite papers only relevant to our results obtained. B is the papers to be cited should be old classic papers that worked on the area for the first few decades. The C is the studies cited should strongly be related to your research question, or D is all of these. Okay, so the answer for this particular question is that the studies that we cite should strongly be related to your research question, right? This means that whatever hypothesis you were providing, the citation should be around that question, right? It could be positive or it could be negative for whatever work you have done. That is why we are saying that we should cite papers only relevant to our results obtained. No. So even if there are contrasting or contradicting papers to what are the results that we have obtained, people are anyway going to find them, right? Might as well cite them and might as well clear up that, okay, these people said that this was the case, but when we have actually worked, we found that this instead is the case with this particular the condition that we are getting might differ between which stage of cancer it is, right? So something like that. So you will also cite papers that might have contradicting results to yours. or the papers to be cited should actually be the current papers, right? The recent things that have been done should be cited. And the exception to this is that whatever was the first paper or the first few papers to work on that particular, maybe you are working on maybe say Duchenne muscular dystrophy. So the first paper ever to, introduce or find out about the gene responsible for DMT or something like that. And maybe you are working on a genetic this thing gene therapy for DMD, right? So the first paper to actually identify the gene for that particular disease should obviously be cited. It might be 50 years old, right? But it should still be cited, right? Because that is how you came to know that, okay, this was the gene, and this is the gene which I have to therapeutically, improve, or the function of which has to be made up by the therapy, right? So that is how this question was to be solved. Question number 18. Which of these should be included in a discussion section? Is it restating the results, or taking home or a take home message, or new results that were earlier not discussed in the paper, or introduction to the research question? So a discussion section should have which one of these? I'll just have water. Okay. So the option that is correct for what should be included in a discussion section is a take home message, right? Because we have already stated the results, we will not replicate the same statements over here again in the discussion section. What instead we will do, we will discuss about them, right? If we said that this was the result, sorry, this was a particular result that we saw over here, what we will say is that it might have meant this thing, right? So we will not just restate those results, but we will include a take home message that, okay, this was the research that I have done for proving my for my working towards my research question, or for proving my hypothesis. These were the experiments that I had planned. This is how they turned out to be, and through these experiments, we think that this is maybe the possible mechanism for so and so disease, right? And then, do we write new results that were earlier not discussed in the paper? No, you will not introduce something new in the discussion section, right? Results is a section that has already been dealt with. Even if you have more results, go back and write it over there, right? You will not introduce new results that have never been written in the paper, right? Then introduction to research question. This is something you do in the introduction part, wherein you are first going through a literature review, then gaps in the literature, then you come up with a question, you propose that question, and then you propose experiments to, solve that question. So that is also not true about discussion section. Then question number 19. What is done in a macro structure revision? So, as you might remember, there are two kinds of revisions on a broad perspective. One is a microstructure revision, the other is a macro structure revision. Which one of these is a part of macro structure revision? Is it revision of the organization, or revision of the sentence structure, or revision of the content, or revision of the flow?
Is it C?
Okay. So it is not C actually. What changed? Oh, did I? What I just saw is that what I have written over here is incorrect in the first place. What is not done in a macro structure revision is actually revision of a sentence structure, right? The all other things, that is revision of organization, content, and flow, are the major things that are dealt with when you are doing a revision on a macro structure view, right? But you will just a moment. So that was my bad actually. I have not written the question properly in the first case. I think that's the confusion. Uh, Three of these is done in case of macrostructure revision. They are organization, content, and flow revision, right? But revision such as what is the grammar, what should the grammar be like, should the sentence structuring be like, how should this particular font be like, what how the heading should be written like, how you should name something. All these are micro level or microstructure revisions, right? So that question was my bad. Okay. Yeah. So, okay, I gave away the answer, but we will go through it. When, which one of these has a financial, personal, or personal interest of belief that could affect his, her, or their objectivity or inappropriately influence, just a moment, or inappropriately influence his or her or their actions, a potential conflict of interest exists. So, which one of these are we talking about? Is it in case of an author, in case of a company, in case of a reviewer? Which one of these? So, since I've already given out the answer, we will just skip to that. So whether it is an author, a reviewer, a editor, a sponsor, a company, whatever, if they have some, if they are performing some research, but they also have some personal interest or financial effect, interest that is, being fulfilled by that particular research, it might be the case that their research is biased, right? So that is when you actually need to declare a conflict of interest, right? So all of these is the correct option for this. Then question number 21 is researchers while writing manuscripts often use the abbreviation et al. Yes, Vikas.
Can we come back to question number 20 once?
20, yeah.
Yeah, here, can you just explain the question actually? When Dash has a
Okay.
financial personal interest of belief that would affect here his, her, their objectivity. Here it is referring to whom? His, her, their objectivity. It is about the writer who is writing the research article right now.
Yeah, so it could be the writer. That is what is meant by the author, right? It could be the people who are financing that particular, author, because for your research you will need some kind of a grant. Now that kind of a grant or that kind of a support, financial support, could either come from a government agency. For example, in India we get grants from SIR, we get grants from CSIR, ICMR, DBT, etc., right? But I could also be working on, say, a security system of some sort, a cyber security system of some sort, and maybe Microsoft is funding my research, right? And whatever research I am doing is, if it is to develop something new, well and good. They are also benefiting, I'm also benefiting out of it, right? And the community in general, the cyber security research is also benefiting from it. But maybe I am doing some kind of a research in which I'm testing a few existing cyber securities, right? In which maybe some daughter company of Microsoft is also under scrutiny, right? In Under those different kinds of companies that I am scrutinizing, right? And maybe in my results, I find that that particular daughter company is giving the best kind of cybersecurity according to whatever things that I have checked in it, and then I'm reporting this. But over there, I will have a conflict of interest also, right? Because Microsoft is supporting my research, and in my research, I am looking at cyber security of multiple companies, and one of them is Microsoft itself, right? So I might feel the pressure to, make a biased decision at some point, or, organize my research in such a way that maybe I perform multiple experiments, but I publish only those that will benefit Microsoft out of this, right? So that will be a conflict of interest. Because they are giving me money, I am doing this, right? So maybe journals will not accept such kinds of publications, right? Because it is not scientifically going to be, corruption free or very sound, right? When you have a conflict of interest, even if in case of reviewer or editor, maybe I am a researcher, right? And along with a researcher, I'm also a reviewer for, I am the editor in chief for one particular journal, and I'm a reviewer in many other journals, right? So I have this, I am in speaking terms with a lot of other reviewers also, right? So maybe I am submitting a paper that is going to a reviewer that I already know, right? And maybe one of the reviewers in that is also me only, right? Or maybe some really close friend of mine. So if they are reviewing my work, that is also conflict of interest, right? They will kind of have of this bias that, okay, let this work be published because this particular person has done that. I know this person from a long time. So that is also a conflict of interest. Okay. So it could be anybody. On a company institute level also it could be possible, and on a smaller individual level also it could be possible. So you understand why we are using there, because companies might also be involved in this. Okay, we will jump on to the question 21. Yeah. Okay. Again, I've given out the answer. So the researchers, while writing manuscripts, often use the yeah, Tejaswini.
Others, ma'am, and others.
And others yet. So the et al over here means and others, because sometimes when we are talking about, for example, I have also previously cited some of the works. Okay, if I'm citing a paper, I will write the name of the first author and I will write et al. Right? That means that there are multiple people in this that have contributed to this work. I am only referring to them by the first author name. Okay. So that is what this means. I hope you have come across this term, right? If not, we can just Google one a research paper and I can give you an example. So that is up to you. Okay. Then question number 22 is research ethics do not include which one of these? So these are very, very mm, like you should be aware of these terms to know the answer also. So which one of these is not included in research ethics? Is it honesty or integrity or subjectivity or objectivity? Yes, Vikas.
It should be subjectivity, I guess.
Yeah, so that is correct, because research ethics means what? That you are providing an honest review or an honest opinion or an honest, presentation of data of whatever you have found during your work on this particular project, right? There should be some kind of integrity, of course, right? That there should be data, it should be backed up by the number of times you have repeated that experiment or the number of animal models you have used. That will basically create the integrity in your research, right? Then objectivity should be there because there are multiple objectives that you are trying to fulfill, right? So there are defined objectives of the research and you have to fulfill that using varied kind of experiments. So objectivity is also there, right? But subjectivity is a very, very user relative term, right? So your views on a paper might be different than my views on that paper or my friend's view on that paper. That subjectivity is has nothing to do with research ethics, right? It is a very, very personal kind of um, demeanor to have on a paper, right? So that brings us to the end of the question answer sessions, at least session. Yeah. uh, Do you guys have any doubt with this particular question answer session? If not, we can just go and do a very, very quick recap about of the this week's experiments. Sorry, this week's lectures that were given by sir. I'm sorry. I'm a little tired. I will just have water and I can resume. Okay, if not, because we did deal with some doubts while we were going through these questions in the first place. So if not that, what we are going to do is we will just quickly go and we will revisit. I'll just remove this also. We will just revisit sir's lectures, right? Just the essential part we are going to read out just to refresh your memory about the same. So, okay. I don't know if my pointer is visible or not. I will not be able to see also, and I'm unable to highlight on this. Okay, anyway. So this is one slide that we have discussed time and again, right? Just a moment. I will maybe present my screen again. I'm not sure if that was required or not, but I will. Mm, yeah. Okay. So yeah, what I was saying is this is one of the screens that we have discussed time and again. It basically talks about how there is an R class view when you write a research paper. Yes, Vikas.
I think nothing is being shared. We are unable. I am unable to see the content shared.
Okay, is that the case for everybody? Okay, I'll share my screen again. Is it visible to you now, Vikas?
Is it visible, ma'am?
Yeah. Okay.
Maybe network issue. Maybe
Okay,
it is visible.
Vikas, maybe you can try joining again if it is not visible because, yeah, yeah,
Now
yeah,
it is
yeah.
visible for me also.
Okay, okay. Good. Yeah, so what we were saying was that this hourglass view of how a paper should be written and which part of the paper should be written first, which should come later. Like the first one that is to be written is the methods, then probably the results, then the introduction, then the discussion, then the abstract, and the last one to be written is the title. These are things that we have discussed in the previous session also, right? After what we can do is we can move on with the slide. And since this week's lecture started talking about results, right? Results and discussions. That is why this particular thing is highlighted. When Sir did talk about results, he highlighted some key contents from the Springer website, right? Springer is a publishing group, science journal publishing group. They have many journals under them. Sorry. I'm so sorry. And they have given some tips about how result section should be written. These include that these say that in the result section, especially, we should simply state what we are finding, but we should not really interpret the results or discuss their implications. Right? This is what we have discussed during our session also today. Then they have also said that as we do in the material and method section over here, also we can use subheadings to separate the results of different experiments. Also, results should be presented in a logical order, okay, rather than in the order in which the experiments were actually performed. We should not really duplicate data among figures, tables, text, right? Because it will be very, very, repetitive, tedious for the same reader to go through the same content in different formats, right? Then we should definitely include the results of statistical analysis in the text, usually by providing p-values wherever statistically significant differences are described, right? Then the results section should also include the following. It should have an introductory context for understanding the results by restating the research problem underpinning your study. It should include non-textual elements such as such as figures, charts, photos, maps, tables, etc., to further illustrate key findings if appropriate. Then a systematic description of your results is also required, highlighting for the reader observations that are most relevant to the topic under investigation, right? Then the page length of your results section will usually be guided by the amount of and type of data that is to be reported, and a short paragraph at end should be given to basically conclude the results section by synthesizing the key findings of the study. So this was the line I had asked you to keep in mind, which I have kind of directly picked from sir's this lecture only, right? Then there were some examples that he gave for each of these points. You have the transcripts for that. You can go through the examples, right? Uh, that is not something we will discuss over here, right? Then the general rules about using non-textual elements in your research paper. In this, you should keep in mind that each non-textual element must have a caption that consists of a number and title, right? Again, these are things we have discussed. Number and title over here are what? That figure 2, mitochondrial fragmentation in neuronal cells deficient for leucorin or malin, right? This is the number that is given. This is a title that is given. And in that also you have subsets like A1 has this nsRNAi, malin RNAi, leucorin RNAi in it. C1 has wild type, malin knockout, leucorin knockout, etc. Right? Then the number of figures, tables, graphs, etc. should in the text should explicitly refer the item. This is also something we have discussed already. Then some of the non-textual element may be okay. The first one also we have not read. If the non-textual element within the text is not adapted from another source, but totally your own creation, you can say so. Otherwise, you must cite where you found the original one, right? Also, if some of the non-textual element may be copyright protected, so you might want to get a permission for its usage, right? Maybe if it has been published in another journal, you might want the permission of the journal as well as the author who made that in the first place to copy that particular schematic, right? Okay. Uh, Then we come to the second one for this week. That is where we talked about tips on effective use of tables and figures in research papers. Right. The first step was to check out your target journal, what your target journal has to say on the issue. Some journals limit the number of tables and figures and so have specific guidelines on the design aspects of the display items. Right. Next, we should decide whether we want to use tables and figures or text to put across key information, right? If our key information is suppose one particular statement, we can use a text, right? But if it is, for example, a signaling pathway that we are proposing, like I gave you an example of earlier, right? In that case, you might want to go for figures, right? If it is a meta-analysis kind of thing wherein you have gone through multiple studies and you have summarized the finding of these studies, and based on that you want to make some observation, you will probably go for a table, right? Then after you have decided to use a display item, choose the display item that best fits your purpose based on what you wish readers to focus on and what you want to present, right? We can focus on a particular part and we can discuss about that. Okay. When we talk about tables, uh, to show many and precise numerical values and other specific data in a small space, we use tables. And to compare and contrast data values or characteristics among related items or items with several shared characteristics or variables, right? Why do we say this? Because for a table, you should have the same columns and the same rows, right? So whatever will go in that column should be like every study should talk about that thing, right? Then only you can make a table out of it. Other, if you don't have common points, you will not be able to do this very effectively, right? Then show presence or absence of specific characteristics. Um, okay. this was a very, very important line actually, that the best way to decide on a format of a table is to review a number of papers and see which format is most useful to you as a reader, right? So once you understand what the reader will understand best, because you are also writing a paper, it has to be read by somebody, right? So if you are keeping that reader's perspective in mind, probably you will benefit the most, right? Because after all, the data being presented is for the benefit of the reader. Then, in case of figures, to show trends, patterns, relationship across and between data sets, when the general pattern is more important than exact data values, then graphs are useful.
ऑडियो आ रहा है क्या? Is audio
Okay.
coming?
No. So it got disconnected from my end for that particular time being. I have connected back using another stable connection probably, and I'll share my screen again. Just a moment. Just stop sharing and share. Okay. Yeah. So I was almost at the end of figures, wherein I was reading the last point, that to present a visual explanation of a sequence of events, procedures, geographic features or physical characteristics also, you will probably go with a figure, right? Um, then this is another statement we have gone through, that figures and tables or other display items are often the quickest way to communicate large amounts of complex information that would be complicated to explain in a text, right? So we know that these are some examples of the same. Uh, and often it is a case that many of the readers that will come to actually read your manuscript or your research paper will only look at your display items without reading the main text of your manuscript. Therefore, it should also be the case that whatever display item you are putting is communicating what you want to, right? If somebody is an expert in your field, right, they would not probably need to go through the entire paper. They can look at the figures that you have given, and they can look at the title of those figures and whatever explanation you have given, that okay, this A1 represents mitochondria that are stained with mitofuscin. This B1 represents the autophagy marker, this is this. Okay, so they will know just by looking at those figures only what is the kind of thing that you are trying to put across in this paper. So your display elements should also be very informative. Also, display items are important for attracting readers to your work. Well-designed and attractive display items will hold the interest of your readers and compel them to take time to understand a figure, and even maybe entice them to read your full manuscript, right? Um, okay. Finally, the high quality display items give your work a professional appearance. The readers will assume that professional looking manuscript also contains good science. That might always not be true, but that is a very common assumption that rightfully so somebody could have, right? Then thus readers may be more likely to trust your results and your interpretation of those results, right? Uh, Coming to this part, we are we did talk about figures, tables, etc. For images, if we are including them in the display item, we have to be sure to include scale bars, then to consider labeling important items. For example, if we are labeling them with different colored dyes, we will write that in that picture only, that okay, dappy in blue, because it is in blue color only. Then whatever is in red color, we will write that over here, right? Then indicate the meaning of different colors and symbols used, which is what I said, right? Then for data plots, we have to be sure to label all the axes. So the y-axis, x-axis should be labeled properly, right? Then we have to specify units for quantities. We have to label all the curves and data sets and use a legible font size. So everything should be visible also, right? It is not for your understanding. It is for the understanding of the person who is actually reading it. So a lot of times, if you guys have gone through heat maps or some gene ontology data, although it is very nice when represented properly, but sometimes in papers, people just represent it in maybe font size uh, maybe 5 or 6, wherein you can't even look at the gene name, right? Which is showing an upregulation or downregulation. So the point is, I can't even look at that, right? So what is the point of me seeing that table in the first place? Not just for the colors of it, right? So it is a little frustrating when that happens. Then there were some examples that sir gave you for the same, right? Uh, these are all those examples. And then we were talking about ethics and image processing for publications, wherein the first one was that figures must maintain their original proportions. When we resize an image, we should make sure to change the height and width by the same percentage. Also, we should be sure that no specific feature within an image should be enhanced, obscured, moved, or removed or introduced, right? For example, in this particular case, something has been removed that can be seen very well over here, right? That should not happen because it was not giving you a good result. You have removed it. Now, that is not how it works, right? Then, uh, if you are splicing together lanes of a gel, you must separate it by a thin black line, and the legend must note that the lanes were run on the same gel but were non-contiguous. That is, maybe they ran differently. That is why you are doing this, right? If multiple stain cells are com composited into a single frame, a border must be drawn around the cells that were not originally included in the field of view, right? So if you are maybe including different kinds of images together, then you should draw borders around them, right? Uh, then there was some talk about schematics in biology, and all these schematics, if you can see, they have so much data in them, right? But if you write it down, it will probably take 2 entire slides, more than that, actually. If I write in a very concise way, it will take 2 entire slides. If I write in a very detailed way, that okay, these were the enzymes that were involved, this was the first substrate, after that this substrate was made, this is the formula for this substrate, etc., etc., it will take maybe 6, 7 slides at the least, right, to explain citric acid cycle. But you can represent it in one slide also. So that is the power of schematics, not just in biology, but in other researches and other phases of life as well, right? This was one of the examples that sir gave, wherein some information was being given to you. Based on that, you have proposed, or you had made a diagram, a representation with sir. Then sir again talked about Springer, and how they had suggested an introduction should be written, and they said that the introduction should provide readers with the background information needed to understand your study and the reason why you conducted your experiments, right? The introduction should answer the question, what is the question or the problem that was studied, right? While writing the background, we should make sure that our citations are well balanced. That is, if experiments have found conflicting results on a question, have you cited studies with both kinds of results, the ones that are favoring your results and the ones that are conflicting or contradicting your results? Both kinds of studies should be cited, right? Then they should be current, that every field is different, but mostly you should aim to cite references that are not more than 10 years old if possible, because then only the whoever is reading your research or your manuscript will be aware that, okay, these are the other up to date things in this field, and it is not a very old thing that you have gone through and based on that you have formulated a research question, because somebody else also might have done that, right? So even with what is currently being done in your area, this question has not been answered, so you are answering this question, right? Then it should be relevant. That is, this is the most important requirement. The study you cite should be strongly related to your research question. So that is actually very, very important. Then you should not write a literature review in your introduction, but do cite reviews where readers can find more information if they want, right? Sometimes you cannot just keep on citing individual papers. That is why review papers are written, because if you want to gain more understanding on a set topic, you can go and you can read that particular paper, and it might benefit you, right, on that, or educate you on that particular topic, or whatever research has been done on that topic, right? Then, yeah, so this was one of the slides that in which sir explained why you should always go from a general statement or from a given that one might be aware of to the specific detail of something, right? So a general statement to the specifics, that is and then the question that is being investigated, right? So having said that, the general phases that are associated with writing in an introduction include establish an area to research by, wherein you highlight the importance of the topic and make general statements about the topic and present an overview on current research on that subject, right? Then you identify the research niche. So it will be a very broad area that you'll be talking about. For example, I'm talking about neurodegeneration to begin with, but I have to focus on one particular disease, right? So first I'm talking about neurodegeneration. I will talk about that in general, right? Then I will focus on one subset of neurodegeneration, which is related to epilepsy, right? Then I will identify a research niche that oppose an existing assumption, revealing a gap in the existing research, formulating a research question or problem, and continuing a disciplinary tradition, right? Then I will place your research within the research niche by stating the intent of your study, outlining the key characteristics of your study, describing important results, and giving a brief overview of the structure of the paper, right? My battery here is, it will survive. Yeah. So, for example, I say I talk about neurodegeneration, I come down to a specific part of neurodegeneration related to epilepsy that might cause neurodegeneration. After that, I will come down to some specific kind of epilepsy and why I want to study that, why, what is the research gap that is existing, and what will be my research question to address that gap, right? Then again, we are coming on these statements that you can go and read, right? Then when we talk about research paper discussions, it should include the study's major findings, right? We should state the major findings, explain the meaning and importance of your finding, and consider alternative explanations of the finding, right? Some things you might have validated, some things you might think are possible, but you did not have the means to validate them. You will still include them, right? Then research context. Compare and contrast your findings with those of other published results. Explain any discrepancies and unexpected findings. For example, we talked about this, that you have to give whatever you are assuming. You have to disclose the assumptions of the paper, and based on that, that these are the strengths of the paper, these are the weaknesses of the paper, which is said, that state the limitations, weaknesses, and assumptions of your study, right? Then we have to close the paper. While that we will summarize the answers to the research questions. So whatever questions that we asked, we will say that we have tried to answer them, because this was the question that we asked, and broadly we have seen that this is what we are getting through our study, which we think will answer this question up to this extent. However, this part of the study could not be validated, right? So that we might plan for the experiments, or it is an open research question, etc., right? Indicate the importance of the work by stating applications, recommendations, and implications. Then we come to discussions and conclusions, wherein we basically answer the question, or answer this question, that is, what do your results mean, right? And then we end the discussion section with a take-home message. Right. And the things that we should avoid is to waste time restating our results and to introduce new results in the discussion section. Right. Then there were other guidelines for acknowledgements, references, acknowledgements also we have discussed while we were solving those question answers, right? For references, you should refer other work to establish origin of ideas, justify claims that you are making, provide a context for your work, and show that there is interest in your field of research, right? That it is not a dying field. There are multiple people who are trying to work on this constantly, and out of that you have come up with this idea, and that's what you want to work on, right? um, then after this, uh, yeah, well, this particular lecture, it talked about how uh, revision of manuscript is done, right? It is written, and then it is rewritten and rewritten again and again until it is, it cannot be rewritten anymore, and it is kind of answering most of the questions that are raised by most of the knowledgeable people in your area of research. And then there were some levels of revision. One was a macro structure and the other was a microstructure level. At the macro structure level, revision includes revision of organization, content, and flow. So the broader picture, the story and the flow of the paper, right? And the microstructure level includes individual words, sentence structure, grammar, punctuation, and spelling, such revisions, right? When we are revising or editing the manuscript draft, right, we have to read and reread our paper. Then we have to discuss with our peers. We have to, take some, this thing, a review or some comments from them also. Then we have to inculcate them, these comments in our paper again, etc. So that is how it is being done, right? So it should not be this precise, but yeah, so there was a brief representation of how a peer review process is gone through. If you are interested in that, you should really look up into this. Then, when we talk about the research and publication, there are ethics which the respect to authorship, which we have discussed, right? That individual who has made a great, significant intellectual contribution to the study, all such individuals should get authorship. They might have contributed differently to the research. So maybe not everybody might have contributed like the most. So the people or the person who has contributed the most will be the first author. The others could be the successive authors, right? Second author, third author, according to how much you have contributed. Then conflict of interest, we have discussed this. Then plagiarism is one of the most common types of publication misconduct, right? When one author deliberately uses another's work without permission, credit, or acknowledgements. This is very, very like uh, the most severe actions can be taken against you if there is even little plagiarism in your research. So this should always be avoided. And if you cite properly, if you are very ethical and you are following all these ethics, you will not face this issue in the first place, right? Then simultaneous submission is something we have again discussed, that we should not, submit a manuscript to two different journals at the same time, right? So that is not recommended. And research fraud, that we should never fabricate data, right? Because we are trying to advance science. That will not happen if we are fabricating data just for our benefit, right? Then this section talked about a research proposal versus a research paper, how the structure of the research proposal could be a little different. And the research proposal is basically a document proposing a research project either from a thesis work or for sponsorship of the proposed work, right? While research paper is a document that describes an original research on a specific topic and the analysis and interpretation on the research findings. So in a proposal, you are basically highlighting your hypothesis and your objectives, right? In a research paper, you are highlighting your results and your like what you gain out of those results, your discussion, etc. Okay. So the flow is like this. First, you write a research proposal, then you try to execute it. Based on that, you find some observations, you analyze those observations, and that results in a research paper, right? Uh, so uh, what are some of the points that a research proposal must address? That it should talk about what you plan to accomplish. It should talk about why you want to do that particular thing and how you are going to do it, right? If the proposal is for sponsorship, then why you are the best person to execute this proposal, because there will be other competing people in the same field of or area of research. Then, when we are writing a research proposal for sponsorship, we are competing with other brilliant applications, which is actually true, right? When we are actually presenting our research work, that is another important and very, very, you know, fear-inducing task, especially in some people, me included. For example, you have to present your research at various levels. You will present some papers by other authors, and you will present your work, like the proposal for your work. In our department, that is done at a department level, right? Wherein you are proposing that, okay, these were the this is the existing literature in my field of study, and these are some of the lacuna research gaps that I have found, and this is what I want. This is my question for from the same, and these are the objectives that I'm proposing to answer this particular question. So we do that at a department level, and there will be multiple places you will have to present, like in your thesis defense, presentations, in conferences, when you defend your proposal. That is what I was talking about, and presenting the progress of work, right? So there will be multiple such areas. So we should know how we want to convey our ideas and enthusiasm, and avoid the pitfalls that could put somebody to sleep. So we don't want people to be sleeping in our audience, right? For that, we should be a little familiar with the audience, right? We should familiarize ourselves with them in the sense that we should know, like, at what level of, uh, science should I, speak? Like, can I speak jargons? Because are these the people who are people from my area of research? In that case, I can use very, very specific terms also. But if they are people from different areas of research, they are not experts in my area of research, right? So I will try to use like very, very simple words so that everybody can understand, right? Then tell the audience members upfront why they should care and what's in it for them. Then we have to convey our excitement. We have to tell our story, right? For that, it should be a very, very well framed, well formed story. We should keep it very simple. These were some of the pointers that sir did list for you from citing Elsevier again. And you should obviously go through them. Then preparing of a presentation, that is PPT, is also a very key task, right? So I do prepare presentation for these NPTEL lectures also that I have. So it is one of the major parts in my life for now. And I know that you really have to plan well. So even for the lecture, for example, I will have something that is prepared, right? Otherwise, what will I come and do here for around 2 hours, right? I cannot just sit and lie around and chat with you guys. Sometimes the audience will be responsive, sometimes they will not be that responsive. So accordingly, I have to prepare, my material. So, yeah, these were points that you can go through again. Uh, tips on how exactly a PowerPoint presentation should be made. And the tutorial session by Sir was actually one of my favorite lectures for the entire this span of lectures, because in that I really could understand the difference when somebody is throwing jargons at you because you don't understand that at all, because you are not an expert in that field. But also when somebody is actually trying to make you understand what they are doing. So it was an eye opener for me, at least. I have stopped sharing the screen. If anybody has any doubt, you are most welcome to address that. As yes, Deepika. Your mic is turned off if you are not aware.
I wanted to say that can we get last year's examinations question papers from somewhere?
I actually don't have access to that, and that access I think has not been provided by NPTEL to any of the tutors, at least. I don't think it is available, because
Would you suggest some guidelines to prepare for the final exam?
yeah, so sir, you have the PDFs for all the lectures. Go through them nicely, okay? And try to understand what is being said, not just go through the whatever lines are being written, right? So understand the implications for that. So even if the question is framed in a different way, you will be able to answer that, right? So that will be my first suggestion.
Are there
Probably
numericals also in the
no,
exam?
so it is not really a course wherein you can ask numericals, right? What kind of numericals do you think will come over here?
Oh,
It is
they were saying the
more of an
alphanumeric. They were saying.
no, I don't think so.
Okay, okay. Thank
Yeah,
you.
it will be either a statement given, you picking up a correct or an incorrect statement, or it will be like something is presented to you and based on your observation skills, some question is asked, which will test what has been taught to you during this course. Things like that mostly.
Okay, okay. Thank you, ma'am.
Welcome. Yeah, so if in case somebody was not already present during the beginning of the session, I did mention that uh, for the past two times, we actually have been having a final discussion session. Like we are given an option to conduct that or not. But so far, I don't have any word about it. So as of now, this is my final session, practice session for this particular course, that is professional scientific communication. I hope you are doing well with your course, and if you are registered for the exam, all the best to you for the same. It is
Ma'am,
possible.
do you have a YouTube channel?
I don't have a very active YouTube channel. I just have one wherein I am uploading these videos because that is required of me through NPTEL, right? So that is all I have on my YouTube
Okay.
channel. These recordings, right? So if you want that, you can go and visit that. That I think NPTEL will also provide you, because I'll share the same link with them as well. So yeah, that being said, uh, I hope you guys had a good time with the course, with Sir's lectures and with the practice sessions as well. and we all hope that you have learned something also. Uh, we might meet you again next week if there is an additional session. If not, that is goodbye from my end. Thank you for having attended the session. I will also stop the recording. Yeah.